Welcome, everyone, to the East Coast Genetic Genealogy Conference. My name is Paul Mayer, and I'm the population geneticist for Family Tree DNA. I recently moved to a remote part of the Arizona desert, as you can probably tell from the scene behind me, so I decided to pre-record this so there wouldn't be any last-minute latency or internet issues. So bear with me as we start the presentation here. Oh, okay. So this presentation is called Unraveling Your Population History with myOrigins 3.0. And let me introduce myself first. So, as I said, I'm the population geneticist at Family Tree DNA. My background actually is not in human genetics, but rather in toad genetics. So, I have a history of working on amphibian genomics and genetics and phylogenetics and applying that to conservation. So, my PhD was on the Yosemite toad in California, where I studied the history and the future under different climate change scenarios for that species using genetic data. So my more recent research has to do with population genetics in humans, including this tool, myOrigins 3, as well as several others, including the Million Mito project. If you checked out my presentation at Roots Tech 2022, I gave a brief overview of what we're doing there, but you can check that out online or on YouTube, and I'll give a link to that in a second. As well as several other tools such as age estimates for the tree of mankind, the Big Y haplotree, which includes the largest collection of full Y chromosome sequences. We've added TMRCAs, or times to most recent common ancestor, for the whole tree, so you can check that out as well, and I'll give a link. The cool thing about working on toads is that they don't seem to care so much about privacy issues or the same kinds of hassles that humans have to deal with. So there aren't too many angry customers. But at the same time, it's a lot more fun working with humans because we get to discover so many more interesting and cool stories about human history and how humans have moved across the planet. So that's a brief overview of what I do. And here are the links that I promised. So if you want to check out more about Family Tree DNA, you go to their website, familytreedna.com. More about this tool, myOrigins 3. We have a couple of blog articles about the release from 2020, as well as a few videos that I put together. And actually, this presentation is sort of an amalgam of those videos plus some more examples. Million Mito Project, a couple of links there, as well as, I didn't put them here, but check out Roberta Estes, who's giving one of the keynotes. Check out her blog. She has a couple of articles about Million Mito Project. And that is essentially the largest mitochondrial haplotree ever produced, and we're working really hard. We're really excited about this. We're going to get that released hopefully sometime soon, but that is kind of akin to the tree of mankind, but for matrilineal descent. So stay tuned for more on that. And then lastly is my own personal research website, so feel free to check that out as well. So this talk is organized into four main parts. The first part is an overview of the tool itself, what goes into it, what it looks like, what the features are. The second part has to do with the methodology that went into choosing reference populations, as well as how the different features are used to estimate your ancestry. The third part is some examples I've put together to kind of illustrate what you can do with the tool and how it can help your genealogical research. And then lastly, a lot of the questions we tend to get have to do with, well, why do I have, and fill in the blank, some weird or unexpected ancestry? And so I dedicated the last part to try to explain just why this is not so easy and why it's not so clear cut, why it's fuzzier than some people might think. So without further ado, let's start the first part of the talk, which is an overview of myOrigins 3. We are an incredibly diverse species in terms of our population history. Family Tree DNA is an extremely science-oriented outfit, and I know that all of you and all of our customers tend to be very science-minded. They're very interested in understanding human history and understanding the details that go along with making these tools and the complexities that go along with them. The science of population genetics and ancient DNA continue to give us surprising twists to our journey. And our journey started some 200,000 years ago in East Africa and has continued across the globe for the last 60,000 years as we've marched across all the continents except for Antarctica. And all the while, we've been mixing with archaic humans such as Neanderthals and splitting, merging, and isolating into new territories, onto islands, new continents, and by culture. Many populations that we think of as, quote, pure are really mixes of previous ancestral populations. If you've gotten a chance to check out David Reich's book, Who We Are and How We Got Here, that's a great primer on just what I'm talking about, that every population you can name or think of really is not pure, but is itself a mixture of previous ancestral populations. For example, Native Americans are a mix of ancestral East Asian people and the now extinct ancestral North Eurasian people from over 25,000 years ago. Given that vast timescale of migrations, let me clarify what myOrigins is meant to reveal about your ancestry. There are three broad timelines and three corresponding products. Ancient Origins includes most of human history from around 200,000 years ago to 2,000 years ago, or the Common Era. Right now it tells you your composition of three prehistoric European populations, but we're planning to increase this to hundreds of ancient populations from around the world, not just Europe. And we'll do so in the near future, hopefully. So that's most of human history up until about 2,000 years ago. myOrigins, which is the subject of this talk, roughly corresponds to the pre-Columbian timescale. So again, it's a rough estimate, but from 2,000 years ago to about 500 years ago. And a new product, which is still TBD, will cover ancestry within the last 500 years. So stay tuned, I might talk about that next year. myOrigins is included in our Family Finder autosomal product. Autosomal means that this is a genome-wide test for the totality of your DNA. So what will you get with it? Well, even in previous versions, such as myOrigins 2, you'd get estimated percentages from populations around the world and a pretty looking map kind of like this. You'd also get shared origins. So if you and your match both opt in, you'd find out which populations you have in common. Let's look at the previous map from myOrigins 2. Previously, if you bought a Family Finder kit, your map would include one or more of these 24 populations. You can see that most of the world is included, but there are many gaps. For example, check out West Asia, and also many populations are very general. In many cases, they span entire continents. Like check out Siberia, and South and Northeast Asia are one big blob there. So let's compare this to our new myOrigins 3 map. You ready? Look at this staggering increase in populations. This kind of granularity at your ancestry is really unparalleled in the major direct-to-consumer market. We're really excited to see people's results continue to trickle in, and let's check out this map in more detail on a continent-by-continent basis. So let's start with Africa. In Africa, we have over a 5-fold increase. We've got 8 specific populations in West and Central Africa that are common in African Americans, and another 3 different groups of rainforest foragers who sometimes show up in trace amounts. We also have the full diversity of Semitic, Cushitic, Omotic, and Nilotic groups of East Africa, as well as Southern African people and the Khoisan foragers. In Asia and Oceania, we have over a 5.5x increase. That includes 9 populations in Central Asia and the Indian subcontinent, as well as 6 populations in Siberia, 8 populations that were previously just called Northeast and Southeast Asia, and 7 populations newly added from island Southeast Asia, for example, the Philippines, but also Polynesia, Melanesia, and the Sahul region. The Sahul region refers to the continent that is now split into Papua New Guinea and Australia, but during the last glacial maximum was one continent. I might be a little biased here, but I think this is the best Native American panel available to date anywhere. It includes Inuit people, as well as North American Amerindian, and several exciting Central American groups such as Mayan. It also has a good representation of South America as well. In Europe and Middle East, where we previously had the best coverage, we also are expanding our population count by over 2-fold, and that includes several different Jewish groups such as Ashkenazi, Sephardic, Mizrahi, and Yemenite. We're also giving more specific predictions for Europe, for example, the Italian peninsula now instead of just Southeast Europe, and 3 different Caucasus populations are now added. Now at the beginning, you might have noticed I had kind of a colorful little painting of different chromosomes, And I don't know if I mentioned that we have something related to colorful DNA. So aside from our 90 populations, which is over a threefold increase in pop count, we're also offering something called a chromosome painter. The chromosome painter, instead of simply giving you percentages, also gives you where every segment of your chromosome originated, painted by a unique color. So if you're 100% pure, like this person from Ghana in West Africa, needless to say, this may not tell you much new information. Sorry. But for most people who have some aspect of mixing, some, different components in their family tree from different populations, like this person from Mozambique, they might get something interesting to look at. So this Mozambican man from Southeast Africa has mostly Southern African DNA, but he has some Congo Basin and some East African influence as well, shown by different shades of pink and red. This person from Ethiopia has mostly Horn of Africa pops, such as Eritrea and northern Ethiopia, but has some Northern African and Arabian influence due to recent admixture between those two regions. If you're African American, you might have 1 to 2% Native American DNA, and this could really help you identify and locate it for genealogical purposes. Some people have mixing that's much more recent in their genealogy. This person is Lebanese European with a Lebanese mom and a Northwest European dad, and you can see every chromosome has orange and blue phased to the correct side. This is called triophasing and uses your parents' DNA to perfect your result. It's a future update we hope to have soon. And there are many interesting mixes of people. This person is my favorite. They have a Finnish dad and a Filipino mom, which I guess you'd call Filipino. I love this. Here's someone who is Romani, and the Romani have North Indian ancestry from sometime in the Bronze Age. So this particular person is from Poland, I believe. So it makes sense that they have more than half of their ancestry from the East Slavic population. But as you can see, the gray segments sum to about 6%, and they have that North Indian ancestry, which is from a few thousand years ago. Very cool to see this. Here's someone who is Sami. The Sami people are Siberian native people in West Siberia and Finland, northern Finland. And what's really cool about this painting is you can see these pink and white segments denoting Siberian ancestry, and you can see that they're on both sides. This means that if you're Sami and you get a Siberian segment, you're likely to get it not just from mom, but also from dad. You're likely to get it from both sides because those particular segments are so fixed, so ubiquitous in the population that almost everyone has them. And finally, here's someone who is Finnish with East Slavic ancestry, but also has this trace result from South China. Now, if you just got a percentage result, you might think, well, it's an error. But you can see it clearly painted there. It's on chromosome 11, and it's a decent sized segment. And this is probably a real South Chinese segment that this person has deep in their ancestry. Very cool to see this as well. Here is Roberta Estes, and she's giving our keynote later, and she's very proud of her Native American ancestry, and she knew about segments on chromosomes 1 and 2, but this third segment on chromosome 13 is new to her and was produced from our myOrigins 3 tool. Someone who is from the Seychelles might have ancestry from all around the world, all different continents, Europe, Asia, Africa, and the Middle East, and it's just a mishmash of these different continent level segments. So regardless of whether you're one population or many, as someone from the Seychelles, you're likely to get something from this new myOrigins tool. So at the very least, we will be considering a staggering 90 populations, which is, considering only percentage tests, pretty much the best you're going to get on the market right now. So upwards of a threefold increase in the populations we consider when we estimate your results. You also get the chromosome painting, so you'll find out which segments contribute to your origins. And also, something I'm referring to as genealogical clues. So not only do you get a painting, but you might get segments that tell you something more specific about how your matches are related to you that can help you investigate further into your genealogical research. So I'll talk about that more in a bit. But broadly, remember that myOrigins is part of the Family Finder product. It's available as a transfer from 23andMe, Ancestry, and MyHeritage. It's also retroactively applied to all customers, even if you have a transfer kit. And any future updates will be applied to your kit. So check for sales, they happen often, and I hope you get this soon. All right, before I move on to methods, I just want to mention a couple of things about this feature. Like I said, we're hoping to introduce trio phasing in the near future. We also want to help integrate these genealogical clues in a different way. So right now there's the chromosome browser, which can show you which segments you share with your matches. And we have the chromosome painter, which shows you your population ancestry. But we want to kind of seamlessly put those together so you can more easily see how they relate to each other. We also want to integrate the Y and mitochondrial haplogroups with this a little bit better to give you a fuller picture of your genealogical research. So with that, let's move on to the methods. If you want a deep dive into the specifics, I recommend checking out our white paper. I put a link to it at the beginning of this presentation. But we're going to give you kind of a higher level overview of how this works and the difficulty behind it. So we've already seen just how diverse human beings are. We've seen that our history is complex from hundreds of thousands of years of mixing, isolation, and more mixing. And you know that myOrigins 3 has a staggering 90 populations and has the most currently available for any major percentage test. You've also seen that your result comes with a chromosome painting. The percentages and the paintings are two different types of ancestry analysis. So let me explain to you how we combine the two. And to explain that, first, let's try to understand what we even mean by population. I keep saying population ancestry. What is a population? We, of course, are using the word in a genetic sense, which might be confusing because it has many different colloquial definitions. Genetically, and this is not unique to human beings, there are two important components. The first is that a population must be cohesive within. And it secondly must have distinction from other populations. It must be distinct. So you can see here the spectrum from populations on the left, which are completely isolated, they're completely distinct, and within they're cohesive. And on the right, there's really no difference between those three groups. They're all the same population. So let's put this into more technical terms. So cohesive, what does that mean? Well, it must be a panmictic group of individuals. And what do we mean by distinct? It must have sufficiently low gene flow with other populations. So again, the cartoon at the bottom shows you the two opposite ends of the spectrum. At the left, three distinct groups of people are genetically isolated. And so each population has panmixia, or people intermingling freely able to marry each other within them. And also each group is of sufficiently low gene flow with the next population. So they're isolated and they're completely panmictic within. So let's kind of flesh this out a bit more. To visualize this, let's see what panmixia means. So it means that every person is equally likely to interbreed with every other person in that group. It's basically random mating with no major preference or bias. You can think of this as kind of the drunk college party definition of populations. Now, obviously, no human group is exactly like this, but the point is that some groups behave more like this on average when you consider many hundreds of years. It's not really one generation that matters, it's the average snapshot of many. So here is a group that is panmictic because every person has an equal probability of intermarrying with every other person in that group. The other requirement is that there's less than one migrant per generation. In other words, sufficiently low gene flow. So what this means is that the average pattern across thousands of years shows just one person every generation mixing with the other population, or fewer. So what then are we trying to avoid when we delimit populations? Well, a group like this, where individuals only marry their next door neighbors instead of mating randomly, this would be very hard to delineate because it wouldn't be clear where the boundaries are. Or a group like this with massive gene flow. This is way more than one migrant per generation. So for the methods, we choose references using a combination of tools, for example, principal components analysis, or PCA. Here you see the unedited genetic structure of all European countries in one plot. This is really, really messy, and there are really no discernible boundaries. In order to find predictable genetic populations, we weed down the samples based on several criteria. First, they have to be unrelated individuals. All 4 grandparents must be verified to be from the same population. Only groups that can be consistently and accurately separated using genetic classification tools are used. And now look at how clean this PCA plot is compared to the last one. We've used all these tools. We've manually removed all the individuals that are between populations, and we're left with good references. It's also important to note that populations exist in hierarchies. That's because modern population groups derive their ancestry from much more ancient parent groups, like I said at the beginning. Here you see all 90 of our reference populations. They're analyzed using a genotype clustering tool, and we can force there to be a certain number of parent groups, for example, 2 at the very top there, or 3, 4, etc. This hierarchical structure is important for myOrigins 3. We organize our 90 populations into 34 superpopulations, or parent groups. For example, here we have superpopulations, Myanmar, Philippines indigenous, and Northeast Asia. Inside each superpopulation are 1 or more populations. For example, in Philippines indigenous, there's Austronesian and Melanesian. In Northeast Asia, there's Korean Japanese and North Han Chinese. And we can represent all of our 34 superpopulations as a sort of phylogeny. There's a major caveat here. No phylogeny can represent human history, since phylogenies are simple bifurcating trees. Human history is much more complicated due to all the ancestral mixing. So this tree is not actually representing our history per se. It's only an approximation of how groups fit into other groups. It's an organizational tool. All right. So now that you have a taste of human population structure and how we choose our reference populations, let's dive into the purpose and the goals of myOrigins 3. There's a trade-off between accuracy and the number of populations, or specificity. If you include more populations, then each pop has more genetic overlap with neighboring populations, which lowers the accuracy. Also, to include a chromosome painting, there's another trade-off. Predicting the ancestry of each small DNA segment is called local ancestry inference, local to the segment of DNA. It requires that small DNA segments are actually distinct enough to identify which population they came from. If they're not very distinct, then the chromosome painting will lower the accuracy and also lower the specificity. So there's a 3-way trade-off here. Our goal was to find that sweet spot in the middle, the compromise. Let's illustrate the challenge of the chromosome painting with a cartoon. So here you have 3 different groups, just like before. If they're on different continents, then they're probably isolated and well-defined populations. Ethnic groups on a single subcontinent have more overlap, more panmixia, and as we saw before, might not be separate populations. But here's the problem for a chromosome painting. The segments of DNA that are unique are few and far between. Those are shown here in color. Many segments of DNA remain similar over time, shown in gray. Geneticists call these shared ancestral polymorphisms. Even for people separated by continents like Africa and Europe, in thousands of years, there are many segments of DNA that don't change much. DNA divergence happens very slowly and in little genomic islands on each chromosome. We can simulate this. Take 3 real groups of people, Russian, British, and Iberian, or Spain. If we capture those islands of DNA that are informative about ancestry, we can separate them easily. Here, 300,000 SNPs is plenty. But with only 3,000, there are a few differences. And with 300 SNPs, or only about half a centimorgan, they might as well be the same population. Therefore, in myOrigins 3, we only do a chromosome painting for groups of people who are quite distinct. We only paint your chromosomes at the superpopulation level. Remember those 34 groups? And that way, we don't sacrifice accuracy. So that's part of the compromise. Okay, so here's the overall pipeline. It looks something like this. We estimate your percentages with what's called a global ancestry inference tool, which the global in the sense means it's your entire DNA. It's all of your chromosomes. So we get your percentages and we use all of your genetic markers regardless of chromosome. That means we're able to accurately predict your percentages from 90 specific populations. Then we estimate your chromosome painting using what's called a local ancestry inference tool. And this uses 34 superpopulations. Finally, we integrate the two into a final estimate and display your results. Integrating the two methods is not only completely novel as a method, but we find it actually increases the overall accuracy, as you'll see in a second. So for example, here's a hypothetical family tree. Let's imagine this is your 4 grandparents, 2 parents, and you. We're only looking at chromosome 1 here for simplicity, but keep in mind that the chromosome painting includes all 22 of your autosomes, everything but your X, Y, and mitochondrial DNA. Due to the randomness of genetic recombination, let's say you inherit 13% purple ancestry from your dad's dad. Purple in this case is a superpopulation. Then your percentages might be broken down further into, say, 10 and 3% at the population level, you see? Here's a more concrete example. A hypothetical person has 3 Ashkenazi Jewish grandparents. In this case, the 4th grandparent is half British and half Irish. The chromosome painting shows only 2 colors, Ashkenazi and West Europe, but the percentages break that down further into British and Irish. Again, this is how we include a chromosome painting without sacrificing accuracy or population specificity. This is the compromise. And it works pretty well. Overall, populations are assigned with a mean accuracy of 89%, and superpopulations are assigned with a mean accuracy of 96%. You can see how most of the bars are on the right side of each plot, which is good. You can see how our accuracy is even better in this confusion matrix plot, and I know that concept might sound confusing. All this shows you is if everything is predicted perfectly, then everything should be on the diagonal line. So at the bottom right, you see the Sahul population. So everything that is Sahul should be predicted to Sahul. Ergo, it should be on the diagonal. So if samples are assigned to the correct group, they're shown there on the diagonal. If they're misassigned, they're shown in red off the diagonal. And again, 96% accuracy at the level of superpopulations. Pretty good. In many cases, the accuracy of myOrigins 3 is about 10% better than myOrigins 2, and nearly 30% better in this one case on the middle there. Okay, so that's all I want to say about the methods. And again, check out our white paper if you want to know a bit more. Now I'm going to dive into a few examples. All right, so this first example is actually from my own genealogy. So this is what I'm calling the known genealogy case. You might have a certain line in your tree that you've figured out or partially figured out, and you have a certain connection with someone who's already in the system, who's already a match for you. Maybe you want to explore that further. You want to find other matches on that same line, or you want to validate it. So here is part of my ancestry. I am 75% Ashkenazi Jewish and 25% German and Swiss. So that 4th grandparent is entirely from Germany and Switzerland. And I have this known deep genealogy connection from northern Switzerland, where I have a 4th cousin who matches me in the database. But what if I want to find people who match the two of us and find other people who are part of this line in my tree? And what if I want to validate that this person is in fact a match on that particular line? Well, here's what we can do. We can look at the segment that we share. We're 4th cousins, and we have one shared segment on chromosome 17. And that's kind of hard to see, so let's zoom in. Well, sorry, we'll zoom in a second. Got ahead of myself. So again, that's the segment, and we'll zoom in on that momentarily. But first, let's look at my chromosome painting. So this shows that, like I said, I'm 75% Ashkenazi Jewish, approximately, and 25% Western European at the superpopulation level. So if we highlight chromosome 17, where I match my 4th cousin, we can put that next to the chromosome painting and look at them side by side. And lo and behold, if you look at the Western European coverage, it overlaps with the segment that my 4th cousin and I share. So this is consistent with us being a connection on the non-Jewish line in my tree. So that's good. No contradictions there. what's really cool is if you use bucketing, and this is what I did. I went to my match list and used bucketing and found people on the appropriate side of my tree, my father's side, that has the non-Jewish part of my ancestry. Then I can find people who are bucketed with my father and also match at the same segment by looking at all of the segment information from my match list. And all of these people share this same exact segment at this non-Jewish, this Swiss location that my fourth cousin and I share. And if you look at the coordinates, the Western European segment starts at around 11.5 million, that's in base pairs. And the segment that I share with all of these people also starts at about 11.5 million, same location. So it's very, very promising that this might be a collection of people who all share the same Swiss line with me. And so you might ask, well, okay, but what if it's by chance? Maybe they all match you in different ways, but it's, you know, on different segments or different parts of your DNA. Well, we can use the in common with tool that Family Tree DNA has. And at the very least, we know that all of these people match each other as well. So this is not definitive, but it's a pretty good lead that all of these people here might be on that same Swiss line, and I should be asking each and every one of them if they could share more information. And, you know, we can exchange information about our genealogies and see what we have in common and maybe find more information about our shared ancestors. So again, this is information you wouldn't have had without the chromosome painting. This gives you additional information and validation that people might be matching you on a particular line of your family tree. Okay, here's my second example, and this is what I'm calling the unknown genealogy case. So imagine the opposite scenario. Imagine you match someone, but you know nothing about how you match. So here's, this is not me, this is a random person. Imagine there's someone in the database who finds a match like this one. And I'm going to ignore this first segment because chromosome 9 in the middle near the centromere, we don't have a lot of information. It's what we call recombination poor spot, so we don't have myOrigins 3 information there. But let's focus on this other segment on chromosome 16, okay? So this person finds a match with someone else on chromosome 16, and they want to investigate further. Well, what's interesting about this location is that they have a Native American segment there. They're mostly Western European, as you can see. They have trace amounts of Eastern European and Middle East, but they have a few Native American segments, including this location on chromosome 16. So they email this match because it's very possible this person matches them at chromosome 16, but on their Western European side. So they ask for more information, and here's what the person emails back to them. Here's the second person's chromosome painting, and sure enough, that person also has a Native American segment on chromosome 16. So this is really cool. They might know next to nothing about their Native American heritage, but they can use this as further validation that however they match, it includes a shared ancestor who was Native American. And when you line this up side by side, this just kind of reinforces that all of the segment boundaries are similar. So if you look at where they match, it's between about 25 and 50 million base pairs on chromosome 16. And if you look at the chromosome painting, it's roughly the same region between about 25 and 50. And for the second person, also between about 25 and 50. So again, it's entirely possible, yes, that they share DNA on the other side, on the Western European side. But when you consider that there aren't that many people who have Native American ancestry in the database, it's much more likely that they share a Native American segment. And this is something we'd like to further explore in the future, offer a way to actually phase results like this so customers can know for sure which haplotype they're sharing. But for now, this is pretty decent evidence that they should be looking for a common ancestor who's Native American. And then they can look at their match list and see that they're fourth cousins, and this can help narrow down just how far back that might have been. So this is really cool. This is, again, additional information that was not available before that now is due to chromosome painting, due to autosomal tools that we're introducing. All right, so let's dive into the last part of this talk, which is called, why do I have blank? Why do I have a particular result? And this is a common question, but now you have a primer on why the method works the way it does, why populations are not perfect the way some people imagine them to be, how they have fuzzy boundaries, and how the method does the best of an impossible situation. It makes compromises to give you the results that you get. So we saw how we define an ideal population, but what if populations are not ideal? What if they don't quite meet our definition? Then what? Let's perform a thought experiment. Suppose you're from Europe, okay? And your population ancestry results are influenced by between 1 and over 2,000 years. Genealogically, you have ancestors from everywhere in Europe. And I literally mean that. 1,000 years ago, which is about 33 generations, with enough mixing, you had 2 to the 33rd power, or 8.5 billion genealogical ancestors. Now, the population size of Europe was only roughly 50 million. 85 billion versus 50 million. That means everyone in 1000 AD who left descendants is an ancestor of everyone in Europe today, or of no one. So, all Europeans have essentially the same genealogical ancestors. Then why are there any genetic differences between Europeans at all? That should be the question. Shouldn't the question be, why don't I have ancestry from everywhere? Well, the answer is because there is finite space in your genome. Most of your ancestors contributed no DNA to you whatsoever. Your DNA is really a random sample of ancestors who tended to live nearby where you are or your recent ancestors lived. This plot, frankly, blew my mind, and I hope it does for you as well. In red are genealogical ancestors going back in time, okay? In blue are genetic ancestors going back in time. And the black dotted line is the population size in the world. So, who are those mysterious ancestors in your family tree if there were more of them than humans on Earth? Well, most of them are duplicates, ancestors who show up in your tree more than once. And yes, this is a type of inbreeding, but don't worry. I have it, you have it, literally all of us have it. It's a simple mathematical consequence of having a finite number of people on Earth. Okay, to illustrate this another way, let's look at this chart that comes on our company's DNA swabbing kit. It's a genealogy of sorts. It's kind of shaped like an onion, and you are shown there at the center with your 2 parents as the first layer, your 4 grandparents as the second layer, all the way back to 6 generations ago. So, who were your ancestors before that? Well, by doing some simulations, we can see that beyond 6 generations ago, most of your ancestors contributed no DNA to you at all. So due to the randomness of Mendelian recombination, 2 neighboring Europeans share between 2 and 12 genetic ancestors, and this drops off exponentially across distance. The average for any 2 Europeans is less than 1 ancestor. And why is that? It's because they share more lines in close proximity. So 2 people who are closer together in space are more likely to have shared DNA simply because there is more gene flow occurring between neighbors. Here's a simulation done by the Coop Lab at UC Davis. On the horizontal axis, you see geographic space. Imagine this to be latitude in Europe, for example. And if I run the simulation from generation 1 to generation 20, you'll see that the genetic ancestors in blue are a random subset of the genealogical ones in red, but you can see how they're clustered in the center because there's a much higher probability, it's not definitive, but a higher probability that they'll sample from where most of the genealogical ones are, which is in the middle. So again, although your ancestors are from all over Europe, assuming you're European, your genetic ones in blue are mostly from the same place as you. And remember I said this all assumes there was lots of mixing. So was there? Yes. Plenty of gene flow that helped to erase population boundaries. This plot shows that on average, there were 50 genetic migrants between European countries. And remember at the beginning I said that it only takes about one migrant per generation to erase those boundaries. There were especially high numbers of common ancestors shared between many Eastern European populations from events such as the Slavic and Hunnic expansions. Only the countries shown in red stayed somewhat genetically distinct over the last couple of millennia. You can see the Italian and Iberian peninsulas and Finland are somewhat red there, showing that they are somewhat distinct from each other. Another way to show this high gene flow is by conducting another principal components analysis of Europe. Genetically distinct populations would show up as distinct clusters in this plot. So again, we have Spain there in the bottom left, and Italy is somewhat distinct, but we see a general pattern here that's not clustering, but rather a gradient. In population genetics, we call a gradient like this across space isolation by distance. It means that there's no strong population structure in most of Europe. The boundaries between groups are fuzzy and somewhat subjective. To put it simply, most people conceive of populations as this: typological, simple, discrete, really existing. But in reality, there's something more like this. Or, put it another way, population structure is not simple. Try and force yourself to stop thinking of it like a country map with nice and clear borders, and more like this. But so long as we can appreciate this complex history, if we can recognize that most segments are correctly estimated, but some aren't, when we remember that similar adjacent populations are more difficult to distinguish, then we can understand our results better. We can expect the unexpected, and most importantly, we can use this as another tool in the toolkit. No matter what you're researching, it's always important to find more than one line of evidence for any conclusion, and to know how certain any piece of evidence is likely to be. All right, that's it for this video. So I hope you'd enjoyed it, and I hope you learned something, and I am happy to take any of your questions. Thank you.